Hi, my name is Michael Glotzer from the University of Chicago. I want to talk to you for a few minutes about a paper that Masanori Mishima, Susie Kaitna, and I published in Developmental Cell in 2002, summarizing work we did together in my first lab at the IMP in Vienna, Austria. In this paper, we used a combination of biochemistry, cell biology, and genetics to demonstrate the existence, conservation, biochemical function, and physiological relevance of a protein complex that we named centralspindlin. We use the term central spindle to refer to a set of anti-parallel microtubule bundles located between the segregating sets of sister chromatids during anaphase. We now know that centralspindlin is a central player in cytokinesis in animal cells. This protein complex coordinates central spindle assembly, it regulates cleavage furrow positioning, it contributes to Rho activation, and recruits factors that contribute to abscission, the process that leads to separation of the two daughter cells. At the time when we began the study, we had identified CYK-4 in the nematode C. elegans. The most notable domain in CYK-4 is a Rho gap domain, yet this protein co-localized with a kinesin 6 motor protein ZEN-4 on the central spindle microtubule bundles during anaphase. CYK-4 was sufficiently well conserved that we could identify the vertebrate ortholog by sequence. The protein had been identified in testis and was named MGC-RAC-GAP for RAC gap expressed in male germ cells, a name we now consider to be a misnomer, but that's a subject for another time. MKLP1, the mammalian ortholog of ZEN-4, had been previously found to localize on the central spindle during anaphase. Our initial studies indicated that the two proteins were interdependent for their localization, suggesting that they may directly interact. The first key finding of the developmental cell paper was that CYK-4 and ZEN-4 indeed interact quite strongly, and they co-IP'd in stoichiometric amounts. We also found that the human orthologs of CYK-4 and ZEN-4 also form a stable stoichiometric complex. The two proteins could associate when produced by in vitro translation. This allowed for rapid deletion analysis to identify the key interacting regions. Each interaction region contained a coil-coil region that mediated homodimerization. The biochemical properties of the complex indicated that the holocomplex was a tetramer formed by the two individual homodimers. Interestingly, the only temperature-sensitive mutations in CYK-4 that existed at the time lay within the regions that the deletion analysis indicated was important for the interaction. In this paper, we reported about a genome-wide screen for suppressors of the temperature-sensitive mutation in CYK-4 and found, quite amazingly, about a dozen mutations roughly equally distributed between the CYK-4 interacting region of ZEN-4 and the ZEN-4 interacting region of CYK-4. This genetic experiment nicely complemented the biochemistry and proved that the interaction was physiologically relevant. Finally, we found that the centralspindlin complex, but not the individual proteins, have microtubule bundling activity. Collectively, these experiments revealed that centralspindlin is a conserved complex that directly mediates microtubule bundling during anaphase. This paper has been quite influential, largely because a number of cytokinetic proteins have been found to directly interact with centralspindlin, or whose localization is dependent upon centralspindlin-mediated microtubule bundling. These include aurora B kinase, anilin, adapters for the escort3 complex, and perhaps most interestingly, the key activator of Rho. This activator is a ROGEF called EC2 that binds specifically to phosphorylated CYK-4. Thus, the centralspindlin complex serves as a signaling center that organizes cytokinesis from start to finish. I want to thank my collaborators, Masanori and Susie, and I want to thank Bruce Barman for suggesting our paper.